Good day, everyone. I'm Jessica Lockwood with EA Engineering, Science and Technology. Welcome to our Microsoft Teams live event. I'm going to go over some housekeeping items with you before we get started. Online attendees are in listen-only mode, and you can ask a question by typing it into the Q&A panel. There is an icon in the top right toolbar. It's a little question mark inside of a chat bubble. For this event, Q&A will be open for the entirety of the presentation. However, the presenter will not be taking or responding to questions until the end. Here is Ivy Harvey with today's presentation.
Thank you, Jessica. Good morning, and thank you everyone for taking the time to attend our presentation. I'm Ivy Harvey, EA's SERDP ESTCP Program Manager, and will be introducing our two speakers. Before I do that, I wanted to give a brief overview of our involvement with SERDP ESTCP and why we value and feel fortunate to be a part of this program. As a longstanding partner with the DoD, EA recognizes the many environmental challenges that lay ahead when it comes to addressing PFAS-related issues, especially given an MCL has not yet been established. There are no clear way to dispose of IDW, and ecological screening values have not yet been established. The SERDP ESTCP program is one of the many ways the DoD is taking on these challenges. As a participant in this program, we believe one of the most critical pieces in solving these problems is the technical transfer of information. That is, making sure the information gathered and obtained during these unique studies does not just end up in technical papers and reports that may get buried and overlooked, but is transferred and applied to solve real-world issues that we are experiencing in the field. At EA, it is our business to work collectively with the Corps on the front lines, investigating environmental issues and providing interim solutions for PFAS, as science is working to catch up with full-scale remedies. We are always looking ahead at how the research we and others are doing can be applied and transferred into the field, including with our current ecotoxicological studies and thermal IDW study, as well as PACE's mobile lab solution, which was developed under ESTCP. EA also recognizes this program is an opportunity for us to partner with both government and academia to solve real-world problems. This is the type of stuff our scientists, geologists, and engineers get very excited about. We are always looking for ways to collaborate and partner with the Corps to address environmental issues. SERDP and ESTCP does a fantastic job through its statements of need and topics in highlighting future risks to the government and the associated data gaps that need to be filled. Feel free to reach out to me if you have any interest in partnering on an idea. We understand the unique and important perspective you all can provide on these projects. With that, Jamie, can you move on to the next slide? I just wanted to give a highlight of the voices you're going to hear today. To kick this off, that was Jessica Lockwood. She is the producer of our live event. I am Ivy Harvey, as I mentioned before. I'm the SERDP ESTCP program manager, and I'm just providing a brief introduction before we get started. And then I'm going to pass it on to Dr. Jamie Susky. She is a senior scientist at EA with 17 years of technical experience supporting ecological risk assessments. Prior to working at EA, Jamie worked for several years as an ecotoxicologist at the EPA in the Risk Assessment Division. Her current area of research is focused on ecotoxicological effects and risk assessment of contaminants of emerging concerns such as PFAS. Jamie participates on the ITRC PFAS team and has co-authored the ecotoxicological section of the ITRC PFAS technical and regulatory guidance document. She earned her bachelor's in science in biology from Towson University, her master's in environmental toxicology from University of Maryland, and her PhD from in ecology from Texas Tech University. After Jamie is done presenting her projects, we're going to hear from Dr. Frank Branco. Dr. Frank Branco is the Senior Vice President and Director of Quality Control at EA. He has over 20 years experience in providing technical direction on environmental projects. At EA, Frank plays a central role in the quality management program as the Director of Quality Control. Frank also serves as a senior hydrogeologist and senior engineer specializing in the investigation and cleanup of traditional and emerging contaminants in groundwater and soil using innovative in situ and ex situ technologies. His acumen relates to the transport, transformation, and fate of subsurface contaminants in the natural and engineered hydrogeologic systems. Frank has a bachelor's degree in geology from Duke University, a master's degree in geology from the University of Texas Arlington, and a PhD in environmental sciences and engineering from the Colorado School of Mines. Next slide, please. So here's a quick overview of the projects we're going to touch on today. Jamie, as I mentioned, is going to talk through the ecotoxicological projects that we have. There's 4 of them that she's going to cover, and then she's going to pass it on to Frank Barranco, who's going to cover thermal treatment of PFAS, investigative derived waste, which is a recently completed project in 2020. And he will also touch briefly on a project that was developed in ESTCP, and although EA was not a part of its development, we are very excited about this. It's PACE Labs, a mobile service lab that we're hoping to use on some remedial investigations. So Jamie, I will hand it over to you.
Thanks, Ivy. Okay, so as Ivy mentioned, I want to talk about the projects that we're working on, but I want to make sure that I conclude them with a statement on more than our academic and theoretical exercises and how they actually would apply to risk assessment and what we are aiming to do. So, go to the next slide, if it will. Okay. So the first project that I want to talk about is, sorry, I'm looking for the little laser pointer. The first project that I want to talk about is one that we've been conducting for a few years now. This is on the ecotoxicity of PFAS and PFAS mixtures to fathead minnows. This is in collaboration with Towson University. There are 2 experimental designs here. The first one is looking at the effects of PFAS over these critical life stages of both reproduction and development in fish. And so the aim of this project is to give us some good information on the toxicity of these chemicals, either as they exist alone or in a mixture, and set us up for longer-term exposure durations. And so some of the few PFAS that we have studied to date are PFAS, PFOS, PFHXS, and then a combination mixture of PFOS, PFAS, and PFHXS, and then finally PFNA. These first 2 experiments have been completed and they are currently in publication right now. They've been accepted. The second 2 experiments, they have been completed and we are analyzing the data right now. This is what our experimental designs look like within our ecotoxicology lab at EA. And right now, what we can say is that so far, what we know is PFAS is the most toxic of the PFAS we have tested. It seems to be the driver of toxicity in most wildlife toxicity tests. For what I have down here is this potentiation comment. And so what we've learned is with PFHXS, we do not hit a threshold of toxicity. So there's no toxicity found in the exposures that we're using, which are pretty high, higher than what you would find that are relevant to surface water concentrations on the installations. But if you combine PFHXS and PFAS, you find toxicity that is greater than PFAS alone. And so what this says is that PFAS is the driver of toxicity, but in combination with other PFASs, we may see greater toxic effects. And so this is some of the preliminary data that we have on the mixtures. So what this is all leading to is the second step, and this is the effects of PFAS, and this is going to be a mixture study as well, on a full life cycle of fathead minnows. And what we're going to do is follow this guidance document from EPA, although dated in 1982, it's still one of the best guidance documents that are available. And so how all of this fits into risk assessment is helping derive these ecological screening level values. And so, for example, what I have up here, this figure is showing a species sensitivity distribution that is developed by Environment Canada. And so on this plot are all of these different toxicity tests that have been conducted, and a lot of these are from the primary literature. And so we plot them out along this curve, and we can develop these effective concentrations at the concentrations where we would set screening criteria. And so where we think we're going to find the results of our full life cycle test are somewhere down here at the lower end of the curve, and so likely going to be influencing these screening level criteria. Our second project that we're working on is looking at bioaccumulation of PFAS, and this is also in collaboration with Towson University. And so what our aim here is to determine how fish physiology and environmental variables may influence the uptake of PFAS from surface water. And our ultimate goal is going to be to predict PFAS concentrations in fish from surface water samples. And so we know that, and the data seem to suggest that PFAS migrate through groundwater and hit the surface water. And this is where fish are exposed. What we don't know is how the different fish physiology, as well as environmental variables such as temperature and salinity within the surface water, can alter the PFAS uptake. And so we're using a combined approach of laboratory and field work to address this question. This is our rather complex laboratory exposure design, where these are the 3 fish species we intend to use within this experiment. So we're looking at smallmouth bass, bluegill sunfish, and channel catfish. We expose them to 1 PFAS mixture that would be relevant to what is found in surface water on installations at these 3 different temperatures and following these 3 different salinity regimes. And then for channel catfish alone, we want to see how the dietary preference may impact the PFAS accumulation. And so we'll couple this with field investigations that are currently underway. One is at the Naval Air Station in Willow Grove, and then the second is at Andrews Air Force Base. And so currently, we've been collecting water samples, sediment samples, as well as biota samples at the recreation ponds on the installation, as well as surface water just outside of the installation. And so what we learn in the field will help feed back information to what we're going to conduct in the lab. And so where does this fit into risk assessment? So PFAS is going to be measured in all of these different compartments within the biota. So within the muscle, the blood, liver, and the carcass. These all apply to wildlife health, which is the focus of SERDP, is on wildlife studies. However, we'll be able to tease out information for human health by also looking at the muscle. So this provides important insights into PFAS bioaccumulation factors of multiple fish species, as opposed to just sampling 1. It also is going to create this ability to model and predict fish tissue concentrations from surface water samples, which is our ultimate goal. And then also provide insight on how environmental variables can alter the PFAS accumulation in fish tissue, because fish aren't living in a vacuum, in a static environment. These things, their environmental variables change that they're exposed to, and that can alter the way that the PFAS are accumulated. The third project I want to talk about is one that we have recently completed, and we're working on a follow-up effort to this. This is a risk assessment of PFAS to threatened and endangered species. This was in collaboration with Willow Applied Ecology, and we had 2 objectives. The first objective was to determine the overlap of PFAS contamination and listed species on DoD installations. And then the second objective was to develop a tiered assessment approach for the listed species screening level risk assessment. So while we don't know what the hazards or the toxicity of PFAS are to listed species, we decided we would approach this from the exposure side of a risk assessment. So are they even being exposed? Do we need to worry about the risk? The sites that we evaluated within this project are on Joint Base McGuire-Dix Lakehurst, and Barksdale Air Force Base in Louisiana, and then Naval Air Station Patuxent River, that's Maryland. This is a simplistic overview of the framework that we employed. We did this simultaneously, determining what the listed species were at each installation. You can also apply this to other wildlife receptors. This doesn't need to be specific to listed species. So you determine who's there, and then you determine where the AFFF release sites were. Now we know that PFAS doesn't stay at those release sites, that it migrates from them. So we extrapolate that migration across the landscape where it ultimately terminates at a surface water body. And so we take these GIS features of the habitat overlap as well as the AFFF release sites and then overlay them together. And so we prioritize now which species may be at risk of PFAS exposure based on our spatial assessment. And then we can take whatever ones would come out as a priority and run them through a probabilistic risk assessment. And we did that with 2 species, with the bald eagle as well as this northern pine snake. So this is a figure showing what the spatial exposure potential model looks like. And this here is for the northern pine snake, and this is of the Lakehurst area only of JBMDL. And so this, the black hash marks, which you see all over the installation, is indicating the distribution. So where the northern pine snake can move. So these little green patches are optimal habitat for the northern pine snake. The black hash marks are where the pine snake can move. And then the red, yellow, or even orange circles are the AFFF release sites. And then the purple hash marks are the migration pathways that we would expect to find following surface water, as well as the topographic features of the landscape and migrating to the surface water bodies. And so the overlap that we see in the purple hash marks as well as the black hash marks with the light green patches demonstrates where we would expect to find higher exposure potential. And so this is just an example of what this spatial exposure potential model looks like and how we're able to prioritize species to see where the AFFF release sites are, where the habitat overlap is, and then we quantify that and prioritize those species. So this is a cartoon framework of what our probabilistic risk assessment looks like that we conducted on both the northern pine snake as well as the bald eagle. And we start by determining, so these are species specific. So we start by determining what the food ingestion is of the species based on their metabolic demands. We then estimate the PFAS in the food items from the impacted habitat, and we ultimately compare that to a toxicity reference value, which currently are coming from the primary literature. So the probabilistic construct of this framework, so if you're not sure what that means, it means that these are the things that are varying within our model. And so what varies in our model are the metabolic demands of the organism, the foraging location, where did it select its food from, what dietary item did it select? Did it select a fish or a mouse? What size is the receptor? And then the PFAS concentration within that dietary item. And so this is what that model looks like from the probabilistic exposure assessment. This is the output of a model for one individual, and we run this 1,000 times to get a good estimate of what these exposures ultimately are. So along the x-axis, this is for a bald eagle and eaglet. is the days since hatch. So up here at 60 days is where we would expect the eaglet to be fledging. And then along the y-axis are PFAS concentrations within that dietary item that it was fed by its parent. And so each time this red line is following those PFAS concentrations in the dietary items. And up here in this dietary item, it was a lot of PFAS that it received. But down here where you see this line kind of flat, it doesn't necessarily mean that the eaglet isn't feeding. What it means is that it just didn't have PFAS concentrations within that dietary item at that time. And so the working avian toxicity reference value that we have for fish eating birds is currently right here at this 0.021 milligrams per kilogram per day. And that's illustrated by this dark line right here. And we can see that many PFAS concentrations are exceeding that in the dietary items. And so our preliminary data from this model suggests that eaglet is likely a sensitive life stage, and there may be significant risk of PFAS to eaglets. So clearly this has direct benefits to risk assessment. And I think one thing that we can take from this is that we're able to identify potentially sensitive wildlife receptors based on exposure potential, just using data that is already available in the preliminary assessment and site investigation by looking at that spatial habitat overlap with areas of AFFF release. Secondly, it allows us to be pretty well positioned to conduct these baseline ecological risk assessments, and we can even do the probabilistic risk assessments when they're warranted. So the fourth and final project that I want to talk about is this project now moving towards the new generation of foams. And so this is in collaboration with Texas Tech and Towson University. And we're evaluating the ecotoxicity and biodegradation of some of the proposed foams to replace the current AFFF foams that are currently in use. And so we have 7 products that have been selected by SERDP, 4 of which are on the market, 2 of which have been developed by SERDP, and 1 is a short chain AFFF that we're using as a reference. And so we're using these standard and non-standard test species, looking at a range of receptors to see what the ecotoxicity of the fluorine-free foam products are. Within the aquatic environment, we're looking at these 3 species, algae, invertebrates, fish, our taxa, these are our species, as well as in the terrestrial environment, we're using bobwhite quail, and for reptiles, the brown anole. And then our biodegradation studies are actually being conducted by EA's respirometry lab, MVitri. And so what you see in these blue with these asterisks are studies that are underway and have currently been initiated. And so some data are pending on this, which is really exciting. And so these are the 4 SERDP projects that I have been involved with, and I will turn it over to Frank now so he can talk about some of the projects that he's been working on.
Thank you, Jamie.
Yeah, thanks.
Just want to make sure that you can see my slide. Let's see. All right. Okay. Thank you all. It's my pleasure to present some information on another sort of study today. As Ivy Harvey indicated earlier, it's really our priority to present some of this sort of information to folks in the industry. The SERDP mandates the presenting of information, and some of this is state of the knowledge information that we're getting out there as quickly as we can, so we appreciate the opportunity to present it to you all today. I am going to talk about one of my studies that I was a principal investigator on, and as Ivy indicated, we recently completed this project. It was part of the 2018 SERDP Awards, in which there were 4 awards that were made to various firms related to whether thermal desorption and destruction could be used to eliminate PFAS compounds in AFFF varieties or in PFAS found within soil IDW and other forms of IDW. So some common occurrences that might be of interest for you all relative to the studies where you have release areas of AFFF from fire training areas and soil that has to be dealt with either from an IDW standpoint as you're unearthing it, or milk pond projects in which there are construction activities and you're having to remove soil from those areas of potential fire training areas or other areas where AFFF might have been stored. So a little bit of an overview of this study. We wanted to conduct a very controlled pilot scale study, smaller than field scale, but what I would call intermediate scale in experimentation to determine whether indirect thermal desorption to actually desorb PFAS compounds, and then followed by thermal oxidation at a much higher temperature could be used to destroy PFAS compounds that were tested within this pilot scale environment. And you're looking at on the figure on the left here, this is a picture, a concept photo of really how our treatment train was organized here. There's a sealed desorber drum that begins the process here, and our soil was loaded into this in batch experimentation and then heated to a certain temperature that I'll talk about in a minute. The volatilized off gas from that dryer was then transported via piping to a thermal oxidizer, which was to oxidize the PFAS compounds at a much higher temperature. Again, I'll go through some details in a minute. The photos that you're looking at in this slide are the pilot scale system that consists of both this thermal desorber and then the thermal oxidizer, which is the rectangular apparatus that's sitting on its side here that you see in both the left hand and the right hand picture. The material wrapped around it is just refractory wrapping to maintain the temperature in the thermal oxidizer. The figure that you're looking at on the bottom right happens to be the mixing of some of our PFAS compounds with soil. We used clean store-bought soil that was reflective of U.S. soil classification system material that was of the SP variety, or clean sand with no organics in it. As we wanted to eliminate the variable of organics in this particular pilot scale effort. And you're looking at on the bottom of this electric mixer, you see fluid in the bottom. That's our PFAS solutions that we're mixing with the sand. We only worked at one portion of the experimentation with mixed compounds that were store-bought reagent grade PFAS compounds that we spiked into the liquid, the water, and then spiked into the soil. So we were working only with known concentrations that we pre-mixed in the sand, so that we knew what mass of PFAS, total PFAS we were starting with, which is quite different than other experimentation that relates to using site soils, where we don't have as great an understanding of the PFAS mass that we're starting with, even with running total oxidizable precursor assay and before actually running analytical results on what the mass is. So again, we're starting with a known mass. We wanted to be able to do that because we were looking at trying to basically balance a fluorine balance on the front end versus the back end of the fluorine concentration, the fluorine mass that was emitted. So moving on, this flowchart explains some of the experimentation that we conducted within the SERDP study. So following from the left to the right, well, as I indicated in the last slide, we worked with spiked PFAS compounds, but we also worked with AFFF as part of our experimentation. I am going to focus mainly on the PFAS spiked soil results today, but I'd be glad to answer any questions about AFFF use within our SERDP study as well. We tested at 2 different temperatures for the experimentation. We knew based on prior work that PFAS compounds will desorb at temperatures at around 300 or just above 300 degrees Celsius, or that's about 625 degrees Fahrenheit. We knew that desorption would start to occur at those temperatures, but full-blown destruction, based on prior work, indicated that we needed to be up at temperatures as high as maybe 500 degrees Celsius, which is 932 degrees Fahrenheit, or as high as about 650 degrees Celsius or 1200 degrees Fahrenheit. So we knew the thermal window that we needed to achieve. We just didn't know from prior work that we could achieve certain destruction removal efficiencies for thermal oxidation that were adequate. We also didn't know what ultimate soil concentration we could achieve at those high temperatures. So again, we worked only at 500 degrees Celsius and 650 degrees Celsius for the experimentation. For a subset of the experiments, we ran detailed analytical on the air or the exhaust from the thermal oxidizer. In order to achieve any kind of fluorine mass balance, we needed to know if we were getting complete PFAS destruction within thermal oxidizer. So there's quite a bit of analytical work that we did on the exhaust after the thermal oxidation process. So again, the desorption in the dryer occurred at 500 and 650 degrees Celsius. The thermal oxidation after desorption in the dryer occurred at 1000 degrees Celsius, and we stayed at that particular temperature for all of the thermal oxidation test runs. So again, based on the experimentation, we took the vapor from the dryer and imported it into the thermal oxidizer. So it's a closed loop system into the thermal oxidation unit. The only other additive in the thermal oxidation process was propane that was used to fire the basically both the thermal desorber to achieve the temperatures and the thermal oxidizer. Propane was used as an additional fuel source, and that comes back into play, and it's important in the end result on where the fluorine resides based on hydrogen that is also supplied from the propane during thermal decomposition. All of the analytical work, both of the PFAS and the AFFF spiked soils, was done consistent with the process related to, the Department of Defense's requirements on QSM version 5.3. Modified method 537 was used for most of the analytical work related to the PFAS. The analytical work related to the fluorine mass balance on the emissions from the thermal oxidizer, we had to use a technique that would measure fluoride in the form of hydrogen fluoride, and that's an EPA method 26A for hydrogen fluoride analysis. Some of the important questions that we were trying to answer in this research to improve the state of knowledge of the industry that you could see on the right-hand side of this flow chart. Can we achieve parts per billion levels of treated soil? That's important from the standpoint of what's out there right now within the EPA and the states as far as what are achievable criteria for unrestricted use of soil. This technology that we're testing here is premised on the idea that once you treat PFAS or AFFF within the soil would be available for use as backfill on the site, or we were trying to reach the goal of unrestricted use of the soil after treatment. To do that, we would have to achieve low parts per billion levels within the treated soil. That's based on achieving EPA soil screening levels, regional soil screening levels for soil, which are roughly 100 or 136 micrograms per kilogram within soil. And then various states are coming up with criteria for soil cleanup for PFAS compounds. And this is changing weekly and monthly with respect to how many states are getting involved in establishing soil cleanup criteria. But most of those levels are in the low parts per billion levels for states or the sub parts per billion levels, depending on the compounds that they're considering for regulation right now. Another question, can PFAS precursors be removed from the soil? So this is the AFFF question. Can we also thermally desorb and destroy PFAS compounds that are emanating from AFFF precursors? Can a destruction removal efficiency for the thermal oxidizer be achieved in the realm of 99.99% destruction removal efficiency? This is important because most states have air permitting criteria that relate to DRE and meeting at least the 4 nines or the 5 nines category. And then lastly, can indirect thermal desorption and thermal oxidation achieve irreversible PFAS destruction? So this is the question of destruction. You know, can we prove within this research that we're not just converting PFAS or AFFF precursors to incomplete combustion byproducts, and this is why we went all the way to a fluorine mass balance within the study. I didn't describe earlier that we only used indirect thermal desorption for the study. There is a form of thermal desorption that's direct thermal desorption. The difference between the two only relates to whether the flame is indirectly heating the dryer or the flame is from outside the dryer, which is indirect thermal desorption, versus the flame directly contacting the soil or the IDW that you're trying to treat, which is direct thermal desorption. Both have been used in field applications. We can talk on any question and answer on the merits of indirect versus direct thermal desorption. For purposes of this study, in order to contain just the PFAS compounds that were desorbed from the soil, we chose to use indirect thermal desorption that would disallow any kind of fuel combustion, compounds from the propane that we would see within the emissions mix for direct fire systems. Moving on to some results now. I just wanted to break this down. There's a lot of information on the slide. This is our one slide on the results of this study. On the top two graphs, you're looking at the information related to how we did as far as soil treatment. So there's information on the top two figures that relate to pre versus post soil concentrations after indirect thermal desorption. And let's just take the left-hand slide. This is at 500 degrees Celsius versus the right-hand figure is at the testing at 650 degrees Celsius. The blue bars represent the concentrations of the pre-treatment soil. So these are the spiked concentrations of particular compounds that were used within the this study on PFAS spiking. So there were 6 compounds that we utilized. These were 6 compounds that were mostly part of the uncontaminated, the unregulated contaminant monitoring role, or UCMR3. So the sulfonic acids, or PFSAs, that we tested included PFAS, PFHXS, and PFBS. The carboxylic acids, or PFCAs, that we tested were PFOA, PFNA, PFPEA. So in case you're not recalling which of what is the chemical structure of the carboxylic acid components versus the sulfonic acid components, I've listed the chemical structure on the left here, the R representing the carbon fluorine backbone of the compound, and then showing the last carbon and the carboxylic acid group, which is the double bond to the oxygen atom, and then the functional group of the hydroxyl functional group coming off that. The sulfonic acid compounds represent the carbon fluorine backbone, this sulfur atom, and the two double bonded oxygens, and then the hydroxyl functional group coming off of it. So in aqueous form, these compounds ionize to their various acid forms, which is why you get the carboxylic acids or the sulfonic acids, which were active within the study because we dissolved these compounds within an aqueous solution. So for the PFCAs, interestingly enough, versus the PFSAs, you could see that at both temperatures we did better on treatment for the PFCAs than we did for the PFSAs. The PFSAs seem to have a greater thermal stability than the PFCAs, but at the 650 degrees Celsius temperature, you can see how positive the results are. Again, the blue bars represent the pre-treatment concentrations. The brown bars represent the post-treatment soil concentrations for those 28 compounds that were measured by modified method 537. And you can see that at 650 degrees Celsius, we met these low parts per billion to sub parts per billion levels for soil treatment. This would meet various unrestricted use state criteria like Texas and Alaska that have had numbers that have been promulgated for quite some time. At the 500 degrees Celsius temperature, we were not successful at meeting Alaska and Texas's state criteria for various PFAS compounds. So it would appear that temperatures more reflective of the 650 degrees Celsius range or 1200 degrees Fahrenheit range are required under very controlled conditions to promote this unrestricted use of the post-treatment soil. On the bottom here, you can see the results of our total mass balance of the fluorine. So what you're looking at in the blue bars represents the total mass of fluorine from all of the PFAS compounds, the 6 compounds that were spiked into the solution, representing just the molar fraction of the fluorine component. And the brown bars represent the total amount of fluorine mass that was emitted from the thermal oxidizer. In these cases, for our fluorine mass balance, the brown bar represents the mass of fluorine from the hydrogen fluoride that was emitted from the thermal oxidizer. So you could see that there were 2 test runs here, and we're showing mass balance on both those test runs. And on one test run, we received an 84% mass balance between the input and the emission. For the other run, we received 116% mass balance on the input in soil versus the emission. So, and you can see there are error bars associated with both of the types of testing on the front end and the back end, and you can see how large the error bars are on the back end, where because of use of Pitot tube for the velocity and flow measurements in the stack gas and thermal oxidizer, that there's a relatively large error bar, which is leading to this degree of error that we see between these 2 mass balances. We consider this very positive result. This is definitely illustrating for our study that we're getting complete destruction of PFAS compounds to hydrogen fluoride, which brings up other questions as far as air pollution control, that you need to have within the stack emissions. But this is proof that there is complete destruction of PFAS based on this mass balance. There is a strong perception that PFAS incineration doesn't lead to a complete a PFAS mass fluorine mass balance. Hence, there is a lot of research that's happening on this. But this particular study, in this controlled setting where we knew exactly what mass was input into the system versus not knowing that hidden mass in AFFF precursors, we were able to show this complete destruction. One other thing I didn't report about the results is that even at the 500 degrees Celsius temperature, we achieved about a 97% removal of PFAS from soil. At higher temperatures, at 650 degrees Celsius, you can see that we achieved about a 99.7% removal of the mass. So concluding a little bit about our study and really how this could be used in an applied setting, we clearly demonstrated with this SERDP study in a proof of concept approach at a pilot scale that we can achieve low level parts per billion levels in soil treated by thermal desorption and thermal oxidation. And this would meet certain state criteria for unrestricted use of that material. We also were able to show that we could achieve a thermal oxidizer destruction removal efficiency of not just 4 nines, but 5 nines that would be helpful in meeting air permit regulations within various states. This would be compliant with any state that relies on a DRE requirement for PFAS removal by a thermal oxidizer. We believe that this technology, thermal desorption and thermal oxidation, may have great benefit to the community that is eventually treating PFAS at sites in soil, like at fire training areas or MILCON projects where soils with PFAS impacts have been unearthed. And we compare this to some of the technologies or options that you have right now. Now, this does not include some of the more innovative on-site treatments like soil washing, like in situ permeable reactive barriers that might be using carbon at this point, or other approaches. This compares the thermal desorption and oxidation, thermal oxidation, to some of the off-site technologies that are being used right now. We still, as an industry, rely heavily on transport and disposal to permitted landfills. We rely heavily on transport and destruction to stationary incineration facilities that are currently under some degree of litigation related to that perception of PFAS incineration, which I do believe will get beyond with more research that shows, like our study, that we achieve thermal destruction of PFAS compounds, and then high temperature regenerative methods for things like carbon media that are being spent at sites after use. Those particular options range in cost of treatment generally between $500 per ton and $1,000 per ton. We believe our feasibility estimates at this point show that indirect thermal desorption and thermal oxidation is somewhere in the range of $300 to $400 a ton, depending on the scale of the project and the amount of volume that's being treated on site. These are on-site treatment costs for a mobile desorber that would be used on site. We're designing for complexities, including understanding when we have PFAS co-contaminants and the necessity for a wet scrubber to remove hydrogen fluoride to address the hydrogen fluoride issue based on how hydrogen fluoride would actually lead to corrosion of metal parts within stacks, as well as hydrogen fluoride being a hazardous air pollutant that we need to address. And we proposed within the ESTCP environment to perform some pilot scale testing at a site using a small volume of soil up to about 5,000 cubic yards of soil. But we're also looking for other sites to conduct pilot scale efforts on to improve our understanding of PFAS destruction as well as the air analytics on the exhaust side. And then, so we'll continue that air analytics work with the EPA to try to dispel the continued perception of PFAS, incomplete PFAS destruction within PFAS incineration. So that's it on the SERDP study. As Ivy said, we wanted to talk briefly about some of the PACE Labs work. Their on-site analysis of PFAS has great merit, and before really launching into what those merits are, I just wanted to say that PACE has, as we've confirmed, is ELAP and NELAP certified. Their process of on-site PFAS analysis is consistent with DoD QSM version 5.3 and table B15. And that their comparison of their work on-site versus off-site analytical is very favorable at this point. So we're very bullish about using this technique, particularly with sites that are undergoing RI, where we can make quick decisions, on-site decisions, in kind of a triad type of approach, where real-time decisions can be made based on the results of the analytical, on stepping out where to conduct additional points for analysis here, and this is just a huge time saving. You're looking at a couple of visuals on the right here that relate to our work at Geophos Field, South Dakota, and particularly where we used environmental sequence stratigraphy to answer some questions about where to sample and how to better understand PFAS RI type complexities. And certainly we intend to use the PACE lab PFAS approach for multiple A&G sites that we have under contract with Omaha. That's ongoing work. So very happy to present some of this information on PACE, and very bullish about some of the work that we can do with PACE. So with that, I'm going to turn it over. Let's see. I think we're going to open it up to questions right now. I think Jess Lockwood will read off any questions that you guys might have, and Jamie and I can address any of those questions. Jess?
Did you hear any of that? Was I
no.
muted the whole time?
Yes.
Excellent. So just a reminder to the folks online that you can type your questions in the Q&A panel. It's a little question mark inside of a chat bubble, and we do not currently have any questions online.
I have one question while we're waiting to see if any arrive. Jamie, I wanted to ask about the eaglet work. You talked a lot about the notion of using sort of the probability of various inputs into the exposure. Have you gone to the level of actually creating probability distribution functions for all of those inputs, and you're modeling those probability distribution functions in their broad range? So you've taken it all the way to PDFs.
Yes. So each one of those variables, so things like dietary selection of their item that they've picked, or the PFAS concentration within the dietary food item, all of those are pulled from probability distributions.
Okay.
So it is a rather complex model that is described on that figure as a sort of cartoon to make it easier to digest. But yeah, every single thing that has a probabilistic construct comes from a probability distribution.
Okay, great. Mm-hmm. I also wanted to mention on my presentation on the last slide, I did not give credit to Chuck Coyle, who has prepared with other folks from SERDP a paper that summarizes the various types of PFAS treatment that are being studied within SERDP. And it's a terrific summary of some information both on thermal studies as well as other methods and processes that relate to PFAS destruction. And if you haven't had a chance to look at that ET&C focus article, I would definitely take a look at that if you have interest in various processes for PFAS destruction.
Excellent. Thanks, Frank. So we don't have any questions online. We've had several people saying thank you and that it was very interesting and well received. So we appreciate that. Ivy, do you have any words for closing?
Yeah, thank you very much, Jess. I just wanted to say, everybody, thank you so much for tuning in and joining us today. We really are excited to be able to share our research with you all. And I want to reiterate that the purpose of this research is to take it to the field level and to actually apply this. That's really the purpose of doing all of this. And so if you have any ideas for research, or if you're interested in knowing more about what we're working on, please reach out to us and let us know. We're more than happy to be involved. We recorded this session and we'll be posting a link that will get this out to everyone. If you want to go back over anything, or if you would like to share this with others, you're welcome to pass it forward. So with that, thank you again for attending.
Thank you very much.
Thank you. Have a good day.